It's a pleasure to be with all of you today. I'm going to ask forgiveness while I take off the jacket. The honor of being here deserves the respect of looking fresh, but uh, it's warm and I'll be more comfortable this way. Uh, Many thanks for welcoming me. It's a crazy time with the world on fire. I'm excited to talk about one of my running interests today, so bear with me. And for those of you online, can you hear me okay? Just to make sure we're doing okay. I'm going to take that as a yes till I see a chat otherwise. So today I'm going to try to talk for about 30 minutes on the Vavilovian series. I'm going to use an example that we've been working on in my group for about 15 years with a range of colleagues. A lot of this is work that comes from partners, and I'm going to start by thanking many of the people who have actually done the work, who are responsible for pushing several of these ideas forward. And I'm going to mostly focus on cool season legumes because chickpeas, lentils, and peas have been some of my good friends for about 15 years. But I want to end by articulating why I'm so excited about mung beans, adzuki beans, and rice beans. So this is my second time in this room. I was here in 2018, I think before many of you may have been here. I had a project with Roland. It was an unusual project, and I'm going to take a minute to explain it because it is filled with contradictions. I am a plant ecologist by training. Yes, I'm the chair of a department of animal and veterinary sciences. And that is because a department head signs papers and says no to the faculty, and the actual expertise is less important. The project I had with Roland was a Russian Science Foundation, Taiwanese Ministry of Science and Technology project. I am not Taiwanese, and although my mother's family was once Russian, I grew up in New York. I was invited to take a adjunct position at Peter the Great Polytechnic and had the opportunity to work with some amazing colleagues in Russia and Taiwan in that project, particularly Maria Samsonova, who's a professor at Peter the Great Polytechnic University, and Maria Vishnikova, who's a long-serving curator and leader at the Vavilov Institute for Plant Resources. Project also included Cheng-Ri Li, a professor at National Taiwan University, and Chao Ti-Ting. That project started in 2017 and ran up until the pandemic. We were fortunate to get US funding from USDA to continue some of our work in mung bean on a project led by R.D. Singh, who's a assistant professor, now associate professor at Iowa State University. And I'm here in part talking about Vavilov from this detour through Russia. Before that, I had worked for several years on chickpea as part of the Feed the Future Chickpea Innovation Lab. This was a project led by Doug Cook, who you can see on the your right-hand side. But this was a large collaborative project that had funding independent to partners in Canada, Australia, Turkey, Russia, and several in the CG system. I will talk more about this project in a little bit. The final piece of different projects I'm going to string together as I talk today comes from a Genome Canada project led by Kirsten Bett, who's a geneticist and breeder at the University of Saskatchewan, and is a partnership with Peter Smikkel, a professor at Pulaski University in the Czech Republic, who I had the pleasure of hosting as a Fulbright Scholar in Vermont in 2018. And two of our trainees, Dr. Azalea Guerra Garcia and Alex Silvestrini, now also a doctor, were a key part of this. All right, so having thanked the folks who did the most work, I'd like to talk a little bit about Vavilov. Now, for those of you present in person, how many of you are familiar with the work of Nikolai Vavilov? Just as a question of curiosity. All right. We're going to have a brief history lesson from Vavilov is my scientific hero. I will sometimes switch between Vavilov and Vavilov. I did not grow up speaking Russian. I don't actually speak Russian, but I have had it corrected that he would have said Vavilov. He grew up in Moscow in a merchant family, but during the Russian Revolution, he had just come back from the UK, where he was a student with William Bateson before the First World War. He took a faculty position at the University of Saratov and was very interested in crop genetic resources. He was empowered by Lenin, the head of the Soviet state in its early days, to expand the genetic basis of resources in the Soviet Union. So he spent his short career, which was ended sadly when he died of starvation during the Second World War, while imprisoned, having formerly been sentenced to death. He, before that happened, had been to all of the continents, learned 16 languages along the way, and built what was the first systematic gene bank. Other colonial powers, US, Great Britain, had been collecting germplasm from around the world. The USDA gene bank in the 1920s included about 15,000 accessions of different crops, and they had been largely assembled by US embassy staff in places like Tokyo or Hong Kong, sending seeds in the mail back to the US, and they would send whatever they got back. In the UK, they had made collections of things like rubber, but had never collected them systematically. Vavilov set out around the world to collect material intentionally and in great depth, and built by the early 1940s a collection of several 100,000 accessions of material of every crop they thought could and might grow in the Soviet Union. In a sign that knowing your enemies is worthwhile, a lot of what Vavilov did was modeled on the US agricultural system, where a set of stations across the Soviet Union, which spans multiple time zones, as well as climates from the Arctic to the top of mountains to Sochi, with a subtropical climate not that different than Taiwan, he had collected material across that range. He was sentenced to death in part for his friendships with scientists in the UK and US. He was also a persona non grata for some of his political views, such as genetics being a real thing. There are two ideas we largely associate with him. One comes from the map I used to introduce this talk, that crops have a few centers of origin. The second is that they have fundamental similarities, and I'll explain both of these. Likely you have all seen this map in different forms. This is a map roughly of Vavilov's centers of origin. These are places in the world where agriculture began more or less independently, starting about 10,000 years ago. So in the Middle East, in the Fertile Crescent, in southern Mexico, in southern China, agriculture began independently. In many of these other regions, it began a little bit later, with both a diffusion of crops from these centers, such as wheat and barley being brought from the Fertile Crescent to the northern part of the South Asian subcontinent, or maize potentially being brought to the Andes, but also the independent domestication of indigenous crops in these regions. So the potatoes in the highlands of the Andes. In each of these centers of origin, and this right here is really the center of my talk today, in every place where people began agriculture, They chose legumes as part of their agricultural system. And this is remarkable. How would people from Mexico to China all recognize the value of legumes as hunter-gatherers? Why would each of these systems, which are often quite different as agricultural systems, have legumes in it? And there are striking similarities of many of these species. Some of that is captured in their names or the history of their names. So in the Vigna species, mung bean, adzuki bean, cowpeas, historically they were put in the genus Phaseolus, the same as the New World beans, Phaseolus vulgaris or Phaseolus lunatus, some of the other Phaseolus species. All right, so part of what makes each of these species similar is they share something we now call a domestication syndrome. In each of these cultivated species, they have a set of traits that make it possible for us as sedentary humans to grow them as crops. So, and this is true not just of our legumes, it's true of our grain crops, rice, wheat, barley, maize. It's also true of many of our vegetables. So let's see, I can get a cursor for those of you who are in here. There is a loss of seed shattering or dispersal. So if a legume, wild legumes usually shatter, even some of our cultivated legumes shatter. When they shatter, the seeds are flung some distance away from mom, and that increases the chance that they survive in a wild setting. But if you are a Neolithic farmer, that shattering means your dinner is on the ground. And if I channel the inner Russian that I have, Tolstoy, the great Russian author, in Anna Karenina, wrote about the Russian serfs who had to follow the harvest of the grain. And all the food they would get is the grains of wheat or rye that they picked up after the harvest. And for Tolstoy, this is the pinnacle of suffering, not having enough to eat because you're using your hands to compete against beetles and mice for grains on the ground. It is much easier as humans to cut our grain, put it in a pile, and then feed from that. Similarly, many of our crops have lost their dormancy requirement. Many wild plants, as their seeds are dispersed, they will not germinate in the first year. They might take 5 or 10 years for some of those seeds to germinate. This is a way for wild plants to bed hedge so that they can germinate at a suitable time after a disturbance, after a fire. If an early farmer is planting a seed and it doesn't come up for 10 years, will they come back to it or not? We don't think they would, but maybe. In addition, most of our crops have more seeds than their wild relatives. They've lost their vernalization requirements. In some cases, they have unique colors. They also usually have a larger seed size. So for Vavilov, he tried to systematize this into a set of laws. The closely related genre would have similar sets of variation in size, in color, in ecological preference, and that we could use this to predict where we might find different crops and wild species that might be useful for agriculture. Vavilov had been trained in chemistry, so he used a word from chemistry. In chemistry, a homologous series is a set of molecules with the same functional group. So an alcohol or a hydroxyl group on compounds of different size. And those groups on the molecules give them similar properties. So in agriculture, a homologous series might be species with a similar domestication syndrome, perhaps the same loci underlying that domestication syndrome. So similar, an allele at the same position on a homologous chromosome that causes the same loss of shattering, loss of dormancy. It might be similar function in a system. And in many ways, right here is the answer to why legumes. We have legumes because of the nitrogen for other crops in the system, and the nitrogen that we consume in the form of protein. But more deeply, there may be shared population genetic processes that make crops similar, and these can give us guidance on where we might find alleles for disease resistance, where we might find abiotic stress resistance, and ways in which we may improve yield. And there may be similar patterns of variation. So I'd like to think this is useful. Vavilov himself thought it was useful because it can help us explain patterns of variation in agricultural systems around the world, and because it can be predictive for finding useful traits. So I'm going to explore these ideas in the group of legumes I know best, and then touch on it in another group. All right. So here are 5 cool season legumes. All of these legumes, peas, or Pisum sativa on the top, then chickpea going to your right, fava beans, and then circling back around, grass peas and lentils. All of these are Middle Eastern crops. They can be found in an area that the Greeks called Mesopotamia, between the 2 rivers of the Tigris and Euphrates, in southeastern Turkey, at northern Iraq, and into Syria. The walls pictured here are from the city of Şanlıurfa, or Urfa in southeastern Turkey. For those of you who had a biblical or sort of great book education, this is the city from which the prophet Abraham was thrown from the walls, and is a holy city in Islam because of Abraham's, as one of the forefathers of Islam, because of his presence there. I'm particularly going to focus on chickpeas, peas, and lentils for reasons I'll explain in a minute. We know that these 3 species were domesticated in the Fertile Crescent because there's a particularly good archaeological record in the Middle East. We have 10,000-year-old remains of chickpea in particular that are well dated. If I pull my cursor over again, in the north center of Syria, in some very well characterized remains. The 3 species have overlapping but differing ranges. Wild peas, Pisum, are pretty widely distributed. Lentils, particularly Lens orientalis, has a slightly more restricted range, but it still goes from the country of Georgia into Jordan. Wild cicer, the wild relatives of chickpea, have a very restricted range, limited to 8 provinces of southeastern Turkey. And this helps constrain where the domestication happened. So I'm going to start by sort of walking through some of these components of a homologous series. These 3 species share a core domestication syndrome. The wild relatives are all dormant. The cultivated is not. The wild relatives all shatter. In chickpea, they don't shatter that much, but they shatter a little bit. In the cultivated, they do not shatter. And the seed size is substantially larger in the cultivated than in the wild. And the wild is more bitter. They have high levels of phytic acid and some other defensive compounds. The cultivated is generally less bitter and sweet, and more prone to insect damage because of that. Second one I want to look at here is shared population genetic processes. And this has been a lot of the focus of my interest for a while. And the reason for this is that narrow range of wild chickpea means that it has very little genetic diversity. In fact, in the early 1990s, when the first molecular map was made for chickpea, there was so little diversity that with allozymes, it was impossible to generate a molecular marker map with allozymes because there's no variation in cultivated chickpea in the US gene bank in the 22 allozyme markers that were chosen. The geneticists at the time had to use wild chickpea to find any that were variable. And part of this, and this remains true today, part of this was a matter of collecting effort. ICRISAT, one of the CG centers, is very proud of the 18,000 accessions of chickpea they have in their gene bank, now nicely duplicated in Svalbard. But until we began working on this, only 18 of those accessions were of the immediate wild relative. So that would be like picking me and 18 other people from upstate New York to represent the future of humanity after a nuclear explosion. We might be lovely, we might be horrible people. It's sort of a moot point because we don't reflect the diversity of humanity. These 18 accessions of wild chickpea don't reflect my wild chickpea particularly well, in part because the Icarta team that collected them were excellent, but they drove north from Aleppo. They stopped at the first Arab-speaking village in Turkey they found. They collected, they went a little bit farther, and they turned around and they went home. It's a collection, but it's not a particularly diverse one. So we set about generating a broader collection. We partnered closely with several Turkish colleagues who obtained permits. We were collecting in 2013 before Nagoya came into force, but with the help of the Crop Trust, the seeds are now available on the SMTA through the Turkish Gene Bank, the US Gene Bank, and ICRISAT. We were able to collect across the full range of wild Sicer of the two immediate wild relatives of chickpea in southeastern Turkey. We were able to characterize that variation phenotypically and with resequencing. We found substantial diversity that expanded the amount of diversity available in gene banks by 2 orders of magnitude, so that future efforts in chickpea could take the diversity from the wild Sicer and start to cross it into cultivated chickpea to restore that variation. We were fortunate to collect in 2013 and 2014 when the Turkish military and the PKK, one of the Kurdish rebellion groups, briefly had a peace treaty. That peace treaty has dissipated, and the humanitarian crisis in Syria spread across the border. So further collecting is really in many ways not logistically possible right now. We've had, but similar work can be done in the other species. So Peter, this is work I am not a part of, has done similar studies characterizing gene bank diversity in wild peas. There's a similar loss of genetic diversity in peas, where wild peas have approximately 10 times more diversity than cultivated peas. And we recently assembled a multi-gene bank collection of wild lentil, including the Carta collection, to characterize a similar loss of diversity in lentil. Part of this diversity, we began characterizing this largely with short read DNA sequencing, RAD-seq or DART-seq. We know, however, that only captures a fraction of the genetic diversity that's available. So if Lucas and I, as 2 men of mostly European descent, likely differ on average by about 1 million SNPs, where I have a C and Lucas might have a G. We also likely differ by 1 million structural variants, where Lucas has 2 copies and I have none. Those structural variants are common in humans. They cause many diseases. They're also common in many plants. So Alex, in his recently completed dissertation, helped build a map of structural variation in lentils, so that it's possible to not just use the primary and secondary gene pool of lentil, but to make crosses more distantly. So this map shows some of the major reorganization events in the wild lentil gene pool that we've become interested in. These changes in genomic structure are a major driver of some of the crossing barriers. And this matters in the work that many of us do, not just in these legumes, but if we want to draw disease resistance alleles from Galapagos solanum, for example, frequently doing so is hindered by barriers. Using tertiary or even quaternary gene pool is difficult in large part because of genomic restructuring events. And as the tools to map these become available, we can do a lot more. So I'm going to put a partial check mark on my second sort of criteria for homologous series. This is in part because chickpea is still a little bit different and being much lower diversity. Third area is whether there's similar patterns of phenotypic variation. So as you might guess, I have a weakness for history, being from I have a weakness for history. And Mendel is another great geneticist. He described 7 traits in peas. And interestingly enough, chickpeas have several of these. One of these is a stay green trait, where the chlorophyll is not degraded in the seeds of chickpeas. And it behaves much like the stay green trait in peas. We found that, in fact, it is an allele at the same locus. So a different mutation of the same gene that causes this. Similarly, the flower color locus in peas is shared in chickpeas. And again, it's a different mutation in the homologous gene. I'm going to put another partial check mark here. A third is domestication loci. So one of the key domestication loci, perhaps the most important for legumes, is whether or not they germinate when planted, the loss of dormancy. And this one, in legumes, they have hard seed coats, so it's a mechanical dormancy, unlike the physiological dormancy of, say, crucifers. We recently mapped this in lentils, finding a couple genomic regions that are largely responsible for this. Peter had previously characterized these in peas. And it looks like, although the pathways are shared, there's a similar activity of phenol oxidase enzymes in breaking down the seed coat mechanically. They are different loci when we line up the homologous chromosomes. And in chickpea, they seem to be quite different as well. Peter is in the middle of redoing this analysis as part of a Czech US National Science Foundation project. But I don't expect the loci, although he's using different populations to generate a better map, I don't expect the loci to move that much or be the same. I'm still going to give this I'm going to give this one a no. And I'm going to jump to the last point, the shared functionality. We like legumes because they fix nitrogen. They associate with rhizobial bacteria, and they can bring their own nitrogen fertilizer. This is an amazing ecological capacity. When the legume family arose after the meteor strike that killed the dinosaurs 60-ish million years ago, legumes became one of the most widespread botanical families on the terrestrial Earth. They're very rare in Arctic systems. They're rare in tidal marshes, but in pretty much every other ecosystem from tropical rainforests to grassland, they are one of the dominant botanical groups. That's because in any of those systems, they provide their own nitrogen. They do this in agricultural systems too. But digging in more deeply to that, do they share microbial communities? This, I don't think we know the answer to, but I'm going to highlight a way that we started to approach this in chickpea. As part of our collection of wild cicer, we also collected nodules, both in the wild habitats and at our partner sites in the US, in Ethiopia, Australia, Morocco. And we then sequenced those rhizobial strains. We found that there are completely different Mesorhizobia species associated with chickpea. In India, in the Fertile Crescent, the different wild relatives have different species on them that associate by soil type. In India, where chickpea is the most important country in many ways for producing chickpeas, chickpeas actually associate with a tropical mesorhizobium that was first characterized in something that used to be called an acacia. But a small portion of the genome, a symbiotic island, has been transferred from the Middle Eastern mesorhizobia to the mesorhizobia in India. The same has happened in Ethiopia, and the same has happened in modern development of inocula. So this cassette can be moved between rhizobia to allow a rhizobia that's better adapted to a particular soil to associate with chickpea. How effective it is as a partner requires further work, but this is, I think, a key pattern. In other rhizobia where the symbiotic island is on a plasmid, this transfer can happen even more quickly. To the best of my knowledge, which is limited, this work hasn't been done in other legumes. All right, so I've skipped over 2 other legumes that may or may not follow this pattern in the Middle East. One of these is fava bean. It had its genome finally published a couple of years ago. This was a technical achievement because fava bean has a 13 gigabase genome. It's not a huge plant, but it has an enormous genome that has so much repetitive element space in it that it took a massive effort to sequence it. Furthermore, fava bean has no confirmed wild relative. There is no wild vicia to which you can cross it successfully, perhaps in part because of that genome expansion. Without a wild relative, it's hard to say much about its domestication syndrome. The other is grass pea, and this may be a plant a crop you've never heard of. In places where grass pea is culturally relevant, it is beloved for its unique flavor. In India, it's frequently used in dals and other dishes. In Ethiopia, it's often the preferred legume of the highlands. The Amara particularly love it. In Spain and Portugal, there's a tradition of using it in a number of dishes. It can be ground into a flour and makes a unique tea cake that before the Spanish Revolution was well loved. Grass pea comes with a particular problem, however. It makes a potent neurotoxin, that causes a disease, ODAP, that causes a disease called neurolatherism that leads to paralysis from the waist down. It is rare in contemporary times to see it, although it is still a real problem at higher elevations in Ethiopia. Franco, the Spanish dictator, famously banned the consumption of grass pea because it was causing neurolatherism during the food scarcity of the Spanish Civil War. We don't have a great archaeological record of grass pea. The oldest archaeological remains are in the Balkans. I suspect grass pea is like rye, which was a weed of wheat and barley until it moved to a habitat where it was better adapted than the crop. And it's just rare in the archaeological record because it was a weed. But it also has a different domestication syndrome because it is just as effectively used as a forage crop as it is a grain legume. And the domestication syndrome for forage crops is quite different, if it exists at all, from grain legumes. So they're simply selected differently, and most of our forage crops, even alfalfa, are not that dissimilar from their wild relatives in terms of seed dormancy, pod shattering, or even their chemical profiles. All right, so I'm going to leave a couple open questions. Do we know a whole lot about whether the same genes are selected in independent domestications? In those that we've taken the time to look for, no, but certainly the mechanisms are similar. There's only so many ways to make a plant lose dormancy or lose its photoperiod sensitivity. Mendel's traits were selected because they tend to be caused by one gene. So they're almost cheating. But there are clear similarities here. The reduction in effective population size. This is something that I think still remains interesting and deserves more work, in part because the mildly deleterious alleles that tend to increase in abundance during population bottlenecks are difficult to remove in breeding programs. Even genomic selection has only moderate effectiveness against them. Final issue, and I'm going to use this to jump to my next brief example, is spatial variation. In wild lentil, we didn't find patterns of variation. We found a reduction in genetic diversity from wild to cultivated, but we didn't find similar patterns of northern or more wet area populations between peas and lentils. But I think this is something worth considering. Are there similar patterns of climatic adaptation in different crops? So, and their wild relatives. So let me jump forward to part of why I'm here. I find mung beans fascinating. And part of why I think they're fascinating is in the genus Vigna, there are, depending on how you count, 9, sometimes people say 10 different domestication events. These are crops that are, many of them are in the World Veg Mandate. Some are not. Bambara and cowpea are African crops with long, fascinating cultural histories. But mung beans, mung beans or green gram, urd beans or black gram, muth beans, adzuki beans or red beans, Creole beans, and rice beans are all Asian domesticates. And they span from the driest parts of South Asia through semi-arid parts of South Asia into much wetter and in some cases cooler parts of Southeast Asia, and then to much cooler conditions in Japan, even being adapted over the last 200 years to the high temperate conditions of Hokkaido. I also pose that this is a homologous series, but one that differs in some fundamental ways. They're all in the same genus. They all look very similar. Many of them are cross-compatible or a little bit cross-compatible with one another. Their population genetic processes are actually quite different. Creole, I'm not sure it's even an allowed name, but Creole bean, Vigna reflexosa pilosus, is tetraploid. The others are all diploid. The time since which they've been domesticated differs substantially. We don't have great dates, but cowpea, mung bean, and adzuki bean are probably pretty old. The others are likely, but maybe not certainly, newer domesticates. We know less about the domestication loci in this group, although I know many of you are maybe working with some who are close to having them. And interestingly, these species have very different climatic tolerances. So I'm hopeful that time will allow us to explore some of these questions. To leave adequate time for questions, I'm going to make sure I thank those who do the hardest thinking. I want to give particular thanks to Peter and to Kirsten, Azalea, and Alex for all of their help in trying to make me think straight, and Peter's energy and creativity. So thank you to all of you. Thank you for the invitation, and I'm happy to take questions. And for those of you online, I will do my best to monitor the chat, which I see somebody is here already.
Yeah, so anyone, any questions? Yeah, you can have a look at the chat in the meantime. Yeah, Lorena and then Martin.
I was just going to start just to have people going. I thank you so much for the presentation. I really enjoyed from the history to the amazing questions between domesticated and wild relatives. As a plant pathologist, I always think about the microbes, and I like that you show that definitely there is a differences on their diversity.
Yeah.
And I was gonna ask if you think that, of course, you mentioned at the beginning that the function, that's why is the domesticated syndrome. I like the name.
Yeah.
So these microbes, we know they help in the function. Do you think that the domesticated crops are losing some functions because of the diversity of those microbes? And one thing we are doing with Elon now is asking the reverse question. If we take the wild microbiome to a domesticated plant, can we restore? Can we help enhance some of the traits that maybe are important for us?
Yeah.
I know we don't have the answer, but I just want to hear your
Those are
approach.
great questions. I think the short answer is largely we don't know, but it depends to some extent on the particular crop. So I will jump out of my preference for legumes and jump to maize for a second, where there's some interesting information. My office neighbor at the University of Vermont, Yolanda Chen, has studied, she's an entomologist, and some maize land races and teosinte, or some teosintes, have the capacity, when attacked by certain insects, to release volatile chemicals that are effective at recruiting natural enemies, the parasitoid wasps that will attack the insect pest. The pathway for that capacity is lost in most modern maize. So there's that type of example, I think, happens where we may know less about particular microbial associations that have similar ecological chains associated with them, where maybe one microbe is recruited that is an agonist against another more pathogenic microbe. I think things like root rots are particularly interesting for that. But for that matter, so I think it's a great question. One of the things that pushed me towards this area when I was a doctoral student doing something completely different is work done by Randy Rodriguez at USDA. And I think he's long since retired, Rusty Rodriguez. He was doing microbiome transplants 20 years ago, where he was moving like hot spring soils at the US Yellow Spring National Park between plants and pots, and showing that tomatoes were able to tolerate hot spring conditions when grown on soils that had a microbiome from those communities. So I think there's intriguing things there, but those experiments are complicated to do, and deciphering a mechanism from them, you know better than I how hard that is to do.
Yes, thanks a lot. And yeah, Lorena, so maybe next time when we're going to collect wild vigna in Taiwan on the beach,
sounds like fun.
We should also collect microbiome communities, extreme environments.
Yes.
So that's a lesson for us. Thanks a lot for your presentation. Really interesting with Vavilov.
Yes.
Very exciting. I had a few questions or reflections. One is related to the maybe in going more practical, is that we now look in the Pacific into also what a kind of potential there is for diversification of diets, including legumes.
Yeah.
But maybe from a cultural point of view, the grain legumes are there might be not a lot of adoption, but maybe there's more potential for the fresh legumes, no? Like
Yes.
the winged bean and the yardlong bean. So I would like to get your view on that, both from a food security point, but also from a domestication point of view, which might have a kind of additional complexity in all the population genetics. Then secondly, I wonder to which extent, if you look at the dispersal of Vigna, we work in Madagascar, as you know.
Yes.
So with rice bean, mung bean, they came from Asia, correct?
Mhm.
India, or maybe through Austronesian, Taiwan,
Yeah.
Philippines, Indonesia. So what I wonder is why has rice bean and mung bean been introduced to Madagascar, probably through Austronesian roots, but the same thing didn't happen to the Pacific, because Austronesian also went to the Pacific. It's so, again, it has to do with food cultures.
Yeah.
It seems that legumes didn't enter into the Pacific widely, while they did in Madagascar from Asia.
There is an absence of legumes on Pacific Islands. That's definitely true. Even in the natural communities, that's a little bit less true. There are certainly, you introduced me to this, but there are a range of Hawaiian legumes that are quite important. And in terms of biogeochemistry of the Hawaiian Islands, the legumes are absolutely critical to the function of natural systems on Hawaii, but the cultivated ones, no. I don't have a great explanation for why some voyages would have legumes and others not. In the case of Madagascar, the addition I would make is that there is an Austronesian sort of Malagasy wave into Madagascar, but also very early contact with groups in Africa on the coast. And those groups have not just the Austronesian sort of Southeast Asian connection, but a much stronger connection both to the East Indian trade from Zanzibar and from the Fertile Crescent, and the crop complex in Madagascar. It has cassava, it has Dioscorea on top of the rice, and that mix is just different. And I have been told that it had there was ecological sorting of where different waves of colonists in Madagascar ended up, but they've been trading food with each other for 1,000 years, and that is different than particularly the Polynesian expansion. The first question I've forgotten in the green box. The green, yes, I want to. So, in American English, we have many quirks. I ask you to forgive us. We're not all the same. Beans are frequently called poor man's meat across the world. This is not just an American expression. We have it. In my region of the Northeast, we have very low pulse production as well. And most of the legume production is either soybean, there's 95, 98% of it in the US is fed to animals rather than directly to people. And much of the rest is forage legumes, alfalfa, or either for hay or for forage. Alfalfa, if hay, clovers, or other legumes, if a pasture setting. Really, the only legumes produced, there's a little bit of dry bean production in the New England region, in New York, but most of it is green peas, French, the French lineage of green beans. They're, again, vegetable legumes. We're a little too far north for yard-long beans, but in terms of diet, it's really very similar to that, or to winged bean. There are traditions in Ethiopia, in Turkey, and India of eating green legumes of the pulses. So Turks will go out and sell green chickpeas as a snack. They're eating them very similarly to how we eat edamame, or how we eat, or really edamame, out of the pod in the green state. And it's a matter of just harvesting early. In pigeon pea, there's also the same bifurcation of those that are harvested green and in many cases, a cultural preference for a larger seed, and those that are dried down to a green legume. In the legumes where those are shared, the initial domestication is probably the same, but there's at least in peas and, well, in beans, we know the genes for the green bean trait. There's an enlargement of the pod and further loss of dehiscence that creates a green phaseolus. Those genes give targets for that in other legumes. So if you were allowed to and wanted to, you could edit that in any grain legume to create that trait, and it should have the same effect. Even without that, likely mutations in these genes either exist naturally or can be generated by mutational breeding if one wanted to do it. There are other traits like this that work. You can create a Nunia bean. It's a popping bean. Chickpea, there's also a single accession in the USDA gene bank of a chickpea that you can pop like popcorn. So you fry it or you roast it, and it pops into a bigger open chickpea. The same can be done with the green traits. You can screen for them to get that. I think the domestication syndrome is just a step further after grain. Although in winged bean, I have grown winged bean. I used to have it in my yard when I lived in Miami. It's an amazing pod, but I'm not sure we know enough about its domestication. It's a good question. I have no idea.
Okay, thanks a lot for the answers. Maybe one more question, Ilan, and then we can also see if there are any questions in the audience, online audience, or just the chat box.
A question to your project now at WorldVeg. You're mentioning about the use of mung bean potentially in the US, and a question, how does it integrate also potentially with vegetable farming? Is there any potential for adoption of vegetables from farmers who are already farming legumes, or maybe even for a diversity of diets? Are there any considerations there? So
one of our motivation, or I was thrilled to get the invitation from Lucas to join a project. Thank you. I'm very excited. One of my motivations is I think it's useful to generate general theories of how crop rotations function, what they do for microbial diversity, what they do for disease, what they do for the nutritional value of all of the crops in the rotation. So I'm thrilled about the ideas that are being generated. In US production systems, there are a couple situations where I think mung bean fits well. Two of them have nothing to do with vegetable production per se. One of them, the most common, and Artie Singh's project was built around this, is maize and soybean rotations are the most common rotation in the center of the US. There's a vast area over which we produce those two crops, from below the Mexican border all the way to Manitoba in central Canada, where maize or soybean are rotated. And it's a rotation, but it's such a short rotation that it misses many of the properties of a rotation. The pests of soybean easily survive a year of maize, particularly when they just need to go one field over to find some soybeans. Some of our current policies might change that, but even with that, because the price of soybeans for US farmers has been at best unstable recently with tariffs and other things. But regardless of that, it is economically viable at this point for farmers to grow maize, soybean, winter wheat. And the prices for winter wheat, for reasons we don't need to discuss, have been better than they were in the US. So that's already a longer rotation. After winter wheat in Iowa, there's not enough time for another soybean crop. There is enough time for mung bean. So it fits in well there. A farmer can largely use the same equipment, and there are growing markets for mung bean. Those cases are sprouts, whole beans as a pulse, primarily for dal. We have many Americans who have cultural backgrounds where those are culturally relevant, and they struggle the demand is currently being met by imported mung beans from Thailand or India. The other use case does answer your question. In my region, where there's not a lot of commercial pulse production, there is diversified vegetable production. And I think shifting to eating the greens is something that actually works quite well and is well within cultural norms, even for people who share my background. And we eat greens of lots of different things. And teaching somebody who's already eating Swiss chard, kale, and collards are a little bit, won't tolerate our climate. But if we eat multiple different greens already and know that the sweet potatoes are more delicious as greens, and they are delicious as tubers, then getting people to eat mung bean greens or cowpea greens is not that hard. We eat pea sprouts all the time already.
Okay, thank you, Erika. Is there any question in the online audience before we close the session?
Chat bot.
Yeah. Any colleagues who wants to raise hand?
Thank you all for joining. I don't see any raised hands. Ram, it is great to see you. It's been too long.
Okay. Any final burning question? If not, then we can. Oh, Ram. Okay. Final one. Oh, one more. Okay.
By the way, thank you for very nice presentation. So my question is maybe not directly related to your talk, but looking your the presentation, I realized that maybe with the whatever the CG centers has been established in between 16 to 18, mostly between so are they based on the principle of center of origin of Vavilov principle? Those centers were created in around the world. Is there any connection?
I'm going to give a slightly different answer than I probably should. I know this is being recorded. In international development, the decision makers in countries are frequently motivated to do good. We frequently want to help, but the decisions we make are also frequently ones that come from the perspective we come from and support things that we were already interested in doing. So I went back to the slide of Vavilov. At the end of the First World War, Russia went into a revolution. Several million Russian soldiers had already died before the Russian surrender in 1917. Then Russia went into 3 further years of civil war that brought in several of its neighbors as well, carried on. At the end of that, the Ukraine and southern Russia, the most arable parts, if in the irony of it all, faced an epic drought for 2 or 3 years. So the level of food insecurity in the breadbasket of what had been the Russian Empire, the earliest Soviet Union, was intense. And some 5 million further people, the numbers are not great because of the disruption of the Russian Revolution, but about 5 million people perished during that with incredible human suffering. The US sent food aid to the Soviet Union despite our pretty well-established and documented history of not supporting communism. And why did we do that? During the First World War, we weren't involved in it for several years, and we were selling food at great prices to the Western combatants, to France, to Great Britain. When the war ended, the boys who survived went home and they went back to farming. The price of wheat from the US went down. Why did the US ship wheat to the Soviet Union in 1922? Well, it propped up farm prices in the US. The CG system was largely funded by the US in its early days, and not every station is in a country that was aligned with the US. Carta, for example, the US and Syria, you can say many things about it, but we were not close in the 60s or 70s in a political sense. The US investment in the CG system fell into what was a rather public US Cold War strategy. The US defunding its investments in the CG system at the end of the Cold War, I think is fairly straightforward if you look at it in real politic terms. So where are the centers? The centers are in places where the impact of the investment was greatest. There's no secret why World Veg is here. There's no secret why Erie is where it is. There's no secret why Simit is where it is. And I don't, Vavilov's thinking had nothing to do with it. The centers were geopolitical investments. And that is not to diminish what any of you do. I admire what you do, and it doesn't diminish it. It doesn't even diminish that the people in the State Department in the 50s or 60s didn't care deeply about food security, but they made their investments from where they were, and they had substantial benefits in the US too. And I'd be lying if I said anything else.
Thank you.
Thank you, sir, for the nice presentation. I'm too curious about that large genome size of the faba beans. Its related species, as per my knowledge, is pea and lentil, no, sir. So they have the genome size of 3.2 and 4 GB, but this has 13 GB. Yeah. What may be the cause?
That's a great question. The short answer is proliferation of transposable element families. The mechanism by which that is released is not particularly well characterized. That is hard to decipher from a whole genome sequence, but grass pea is in the middle with an 8 gigabase genome. There are lots of ways to offend people. Geopolitics can be a good starting point, and my apologies for that. But one way to offend people who work on lentil and peas is to tell them that they should all be in the genus Vicia, not Pisum, not Lens, or that they should be in Vicia and Lathyrus and not Pisum and not Lens. We are unlikely to get rid of those names, but from a purely taxonomic perspective, we either need more names for those genre, or we need to call them all Vicia and Lathyrus. Lens is nested clearly in Vicia, and Pisum is clearly nested in Lathyrus. But across the broad group, there are presumably multiple cases where transposable elements are released. Even in the cultigen, even in chickpea, there actually is a little bit more transposable element movement in cultivated chickpea than in wild. And haven't seen any more recent data from my colleagues who were working on this, but likely this is a matter of the bottlenecks during domestication may have inadvertently led to the release of some of the suppression of transposable elements. Similar things have likely happened over the last, say, 5 million years of the Pleistocene and Pliocene, where bottlenecks as communities have moved with repeated glaciation cycles, have likely led to bottlenecking of particular populations or species of Vicia and Lathyrus, and that may have led to conditions that led to transposable element release. I can't prove any of that, but
I have a follow-up question to that one. According to homologous series of Avilo, the same character we find in the crop should be found in other related species also. Yeah. But why we don't find this kind of transposable elements, which is causing for genome size in other related species to faba? And in Chile also, we find it is a huge genome, large genome of 3.5, but its related species tomato is not that much genome size, it is less.
Yeah, it's a good question. I don't have a.
Thank you.
I'll leave it for somebody smarter to investigate.
Thanks.
Okay, yeah, I think we're a little bit over time, so I would like to thank Eric for lots of interesting diversity discussions, enriched by lots of history also. Thanks for sharing all that. And I also want to thank Ram Nair for that, who was the one who linked us with Eric to start this collaboration. So, okay, final comment from Martin, and then we can thanks to Eric.
Just for all the people here from Taiwan and all the people who live in Taiwan. So Vavilov also actually came to Taiwan, correct? Probably a week or so. I don't know how many days. So, I mean, he went across the world, including Taiwan. So just for your records.
Yeah.
Thanks for the comment.
Yeah.
Okay, with that, closing the
Thank you for a great talk,
Please.
Eric. Thank you.
Oh,
Hello, Ron.
final
Good
reflection.
to see you.
Thank you.
Thank you.